Time to get started. I'm Linda Ercole, and I'm the interim director of the UCLA Longevity Center. And I want to welcome you all to our webinar today, which is going to be on the basic links between aging of the brain, the retina, and Alzheimer's disease, risk, prevention, and biomarkers. And I'm happy and proud to introduce Dr. Sally Frautschy. She's a professor in residence at the Department of Neurology and Medicine at the University of California, Los Angeles, and her research lab is at the VA Health Center in West Los Angeles. So Dr. Frautschy has a strong background in physiology and neuroendocrinology and has studied the pathways of Alzheimer's disease now for over 2 decades. And her research involves really understanding these pathways and applying her research findings and translating them from the bench to the bedside. So without further ado, I want to welcome Dr. Frautschy.
So thanks a lot for inviting me. So the first point I want to get out of the way is the disclosures. I have VA and NIH grants, and UCLA and VA have patents of formulation of curcumin, which my husband and I, Greg Cole, are co-inventors. Okay, so I'm gonna go over this through the whole talk, but I'm just giving you a synopsis of the talk and why I'm talking about Alzheimer's disease and macular degeneration at the same time is because of overlapping mechanisms. So one of them is, for both of them, is aging is a big risk factor. The pathogenesis involves both tau and amyloid, 2 proteins that I'm going to be talking about and defining, which is there are several amyloid proteins in a part of the eye called drusen, and I'll be going over what amyloid means and why amyloid associates with aging. They both have oxidative damage. They both have similar inflammation pathways, specifically a subtype of inflammation called complement. And they have similar vascular defects, especially with wet macular degeneration. So on the right, we've got the same type of factors: aging, A beta, tau, oxidative damage, inflammation, blood-brain barrier damage, and vascular disease. So a quick primer on Alzheimer's disease. It's age-related progressive neurodegenerative disease with global cognitive decline. And its definitive diagnosis is neuropathology postmortem, but pre-death, you diagnose via neuropsych testing and imaging. Imaging shows shrinkage, and you can also look at glucose metabolism. With dementia, you get hypo, meaning low metabolism. A lot of people think that Alzheimer's discovered it, but actually Oscar Fisher discovered it and found similar results to Alzheimer's, and that was the early 1900s. Important take-home message is that ApoE4, it's a apolipoprotein E4. It's one of the proteins in the blood that carries cholesterol. It's the most common risk factor for Alzheimer's, and 25% of the population has it. And in clinical trials, half the people in clinical trials with Alzheimer's have this ApoE4. You've probably heard of dominant Alzheimer's disease, where you get Alzheimer's in the 30s. And those are called presenilin mutations. And that means that even if you have one copy, you're going to get Alzheimer's, and you're going to get it early. The most common environmental risk factor is type 2 diabetes. And as I said, it's diagnosed definitively postmortem with aggregated proteins that are the proteins in the lesions called tangles and beta amyloid. So what is an aggregated protein? Proteins fold in 3-dimensional structures. in order for them to function in your body, they have to fold correctly. But throughout your lifespan, including in the fetus, there are always going to be proteins misfolding, and you have machinery to refold them, get rid of them. It works really great until you start aging. And then you call the proteins misfolded. And the most common type of pathogenesis, the disease-associated misfolding proteins, are proteins that form what's called a beta-pleated sheet, which you can see in the background. I have a picture of beta-pleated sheet. It's a way the proteins fold, and it causes disease. So I'm going to give you some examples of proteins that cause disease. So in Alzheimer's disease, the name of the proteins are called tau and beta amyloid. So there's at least 2 amyloid proteins in the Alzheimer's brain. In Parkinson's disease, the most common protein that forms amyloid is alpha-synuclein. In cardiac amyloidosis, the protein that forms amyloid is transthyretin. In myeloma, their antibodies form amyloid. And in macular degeneration, there's a whole bunch of amyloids that are forming from beta amyloid, tau, vitronectin, transthyretin, and there's a whole bunch of others. So there's no other disease like macular degeneration that has so many different types of amyloids. And here's a picture of some of the amyloids in the eye. So the amyloid here is stained green. These amyloid dyes, one of them is called thioflavin S. They're fluorescent, so fluorescence is green. And these are 4 different patients with macular degeneration. And the dyes are not like specific for certain protein, like they're not specific for tau or beta amyloid, but they're dyes that are measuring the beta-pleated sheet conformation, which is disease causing. In Alzheimer's disease and in macular degeneration, you have accumulation of beta amyloid. In the early 1900s, they identified this with silver stains. These top 2 pictures are pictures of silver stain by Alzheimer's, Alzheimer's. These are from his paper. So on the left is beta amyloid. So this is it's neuritic plaque, which has beta amyloid in the core. And then it has a halo of amyloid. And then these branch-like sticks, those are pieces of neuron processes. So these are both the same stain, but on the left, it's staining beta amyloid. On the right, it's staining tau. And these is silver stain for tau. And there's different types of tangles. But the difference between these 2 lesions is that this one on the left is outside the cell and the tangles are inside the cell. And here, these are, this is just like pictures of the nerve cells. I'm going to show you another example, but you can see the tortuous branches here. So that's one of the characteristics of the dysfunction of the nerve cells, because the memory loss doesn't correlate with neuron loss. It correlates with loss of connections. And this graph is just to show you sort of it's pretty oversimplified, because what happens in different regions, whether this exactly represents the sequence depends on what region you're talking about. But mostly amyloid starts early. And if you're in your 30s, you already have amyloid, and it progresses over time. A little bit later, your 50s, you get tangles, tau. And during this time, sort of your 40s and 50s, your inflammatory system starts dysregulating. So inflammation, when is essential for life, it's essential for cleaning up debris and homeostasis, like keeping you healthy. But when it's dysregulated, it causes disease. And in pretty much every single age-related disease, you've got abnormal inflammation. That happens very early in your 50s. And then once you have the onset of symptoms, that represents the loss of connections between cells. So this is the picture I was going to tell you about the branches. So the nerve cells have branches like trees. And from left to right, you have middle age, older, very old. And with the Alzheimer's is on the right, where the main thing that you see, and I guess this was found out with silver stain, which is a way to visualize these branches. But you basically have fewer small branches with Alzheimer's disease, and then fewer branches altogether. So here's a picture of an eye to show you that the macula is in the center of the retina. Age-related macular degeneration is an irreversible destruction of the central area of the retina called the macula. And the retina is the light-sensitive layer of tissue that links the back of the eye and transmits visual information via the optic nerve. Macular degeneration leads to loss of the sharp, fine detail vision required for reading, driving, recognizing faces, and seeing the world in color. It's a leading cause of vision loss and irreversible blindness in Americans age over 60. And advanced AMD is, which is the wet type, is a leading cause of irreversible blindness. As many as 11 million Americans have some form of it. So dry macular degeneration is the less advanced, more common. So 85 to 90% of the cases are dry. This is yellow protein deposits called drusen. And this drusen has a bunch of different types of amyloids that I described before, including tau and amyloid and vitronectin. The photosensitive cells of the central area break down, causing progressive loss. With wet macular degeneration, you have more vascular involvement. The blood vessels are abnormal, and they grow behind the macula, and the photoreceptors die. The vessels are very fragile. They leak fluid and blood. So it kind of breaks down the blood-brain barrier. It causes scarring, and straight ahead vision is particularly impacted. So this wet macular degeneration overlaps with what's mixed dementia. So there's a lot of overlaps between these two diseases, and this cerebral vascular disease with Alzheimer's is called mixed dementia. And it's the most common type of dementia because Alzheimer's disease is almost never pure Alzheimer's disease. There's usually other factors called comorbidities. And with cerebral vascular disease, you get, if you've ever had an MRI, you know, you might be told that you have increased white matter hyperintensities, which could be microbleeds. It's indicative of cerebral vascular disease, and it happens in the white matter. So that's his classic cerebral vascular disease. And if you have imaging for Alzheimer's disease, then they would call that mixed dementia. And it has the same risk factors as macular degeneration: hypertension, high LDL, high HbA1c, type 2 diabetes is a risk factor. And this just shows you exactly how many people have mixed dementia. It really depends on how you define it, because there's not a clear-cut definition of mixed dementia, because it depends on how strict the pathologist is in saying how much vascular disease you have is called like severe enough. So anyhow, there's not consensus on diagnosis of it, but at least 30% of the people with dementia have that. And so we're going to be talking about inflammation, and the inflammatory cells of the brain are called microglia. And dysregulation of microglia, it causes most neurodegenerative diseases. That's a picture of the microglia. They're they're pretty bad beasts. Like they're really important when they behave properly, cleaning up synapses, getting rid of synapses that you don't need, getting rid of debris in the brain, just keeping you healthy. But when they're angry, they cause havoc. They also have different shapes. So when they're doing their business, they get round like macrophages, and they play many of the same role of macrophages in your blood. Here's a picture of some of them from a mixed dementia model, where we have rats that have both vascular disease and they have Alzheimer's plaques and tangles. So on the left, you see a normal aged rat, and that's how healthy normal microglia look. And if you introduce the Alzheimer gene, you see that they're bigger, they're fatter, there's more of them. And then if you introduce only hypertension without Alzheimer's, they're also abnormal. But when you have both, you see it changes their shape, and they're very impaired. And you don't have to worry about that. This is just quantification of those findings. So in the prevention of the current state of the field is mostly treating the people with macular degeneration with antioxidants, lutein, zeaxanthin, vitamin C, vitamin E. You can buy these at these combinations that have been tested and approved by the FDA. You can buy them at any drugstore. But they don't cure the disease. They just slow the progression. So the next 5 slides, I'm going to be talking about curcumin. Curcumin is a molecule in turmeric root. It's related to ginger, and it has this a bunch of interesting qualities. One is that it can bind those beta-pleated sheets I was talking about. So it binds all the different amyloids, no matter what the protein is. The structure of it, it binds amyloid. So that's one interesting thing about it. The second interesting thing about it is it lights up. So it can be used as a biomarker to find, you can put curcumin on a brain section and it will light up all the amyloid proteins, or if you can put it on an eye section, it will light all the amyloid proteins up. So there's a couple more interesting things about it, is how it corrects inflammation. So it's not just anti-inflammatory. It causes, it improves the function of the microglia so they behave properly, so they can clear things better, and it stops abnormal inflammation. And finally, it's a better antioxidant than vitamin E. So in this study, we're using curcumin in patients as a biomarker. So live patients, they're being scanned, and these red dots show where the curcumin lit up. So this colorized image is from this image, where the curcumin labeled plaques in the eye are white. And in this study, they were trying to figure the optimal day after taking curcumin, where they saw the brightest, the best imaging to predict Alzheimer's. And so we went up to day 10, but at day 2 you could see something. So eventually they decided that day 4 was probably the best. But of course, like, it's not that practical for if you have to take curcumin for 4 days and then go to the eye doctor to see if you have it. This is a graph that shows shrinking of different parts of the eye. The fovea, when you have macular degeneration, the fovea goes from 0.06 to 0.04. With as you watch degeneration, you progress over 6 months. So the parafovea goes from 0.03 to 0.01. and the para shrinks, but the parafovea doesn't. And yeah, so then the macula just shrinks. So this is a graph on somebody, a clinical trial done with curcumin, where it shows responders and non-responders. So over a 6-month period where people were taking curcumin, and 10 people were responders. They got improvement. 3 people got no progression. And then there were 6 people who did not respond at all. So we don't know if they have a different genetic risk factor. So we don't know why these people didn't respond and that these people did. So this is a picture of the turmeric root. And I kind of went over that, but what I wanted to do is show you what it does in the brain. So if you feed mice that have amyloid plaques curcumin, let's see, you feed them about the dose that they get in the Indian diet, like the amount of curry that people eat every day. They also put turmeric in their milk, stuff like that. But so you give these mice about the concentration, the human equivalent, and you'll notice that curcumin, so it reduces overall the hyperinflammation that you see away from plaques. But in plaques, the normal way that microglia behave to normal plaques is they cluster around the plaques, but they don't go inside the plaques. So they can't eat and get rid of the plaques. But what curcumin does is it increases the clearance in plaques. So it's both pro and anti, pro good inflammation and anti bad inflammation. As I told you before, like curcumin is shaped like an amyloid binding dye. Like Congo red is a classic amyloid binding dye. It gets into the beta pleated sheet. Curcumin G is another one, and curcumin, it has very similar structure. You see these 2 rings are key, and the distance between the 2 rings determines whether it's going to bind that bad beta pleated sheet in the amyloid proteins. And this is an animal fed regular diet, and these little brown dots, those are plaques. After being fed curcumin, you see the plaques are almost gone. So this has been replicated by many other labs. And in rat models, different models, this bottom panels are just reflecting reduced bad inflammation. So bad inflammation is measured by interleukin-1 beta and tumor necrosis factor alpha. And this graph here, this is measuring oxidative damage. And this is measuring amyloid. So, somebody at Harvard, Brian Baksai, he has this other way to look at clearance of plaques from mouse brains with amyloid, and he makes a little window in the cranium, and he can watch day by day what happens to the plaques. And here he's seeing like plaques around. You can see these red is the amyloid. So you've got red on the vessels and red on plaques. And it turns out that after 7 days of feeding the animals curcumin, that a lot of the amyloid was gone. So that was surprising that it took such little time to get rid of the amyloid. So there's various clinical trials going on with curcumin. Drusen this one is done in Chicago. Drusen contains amyloid protein, which is inhibited by curcumin, a natural plant-based antioxidant. So this study looks at the duration of oral curcumin supplementation to see the effect on reducing volume and number of drusen and decreasing choriocapillary density, loss of flow impairment. So patients will be given curcumin for 12 months, and they'll be evaluated every 3 months. So that's an ongoing trial. The macular damage, and also in Alzheimer's disease, you get what's called innate immune response, and it's primarily mediated by complement system. This special type of inflammation called complement inflammation, you don't hear about that too much. You don't hear about drugs or anything, because you see it's like essential for life that the complement system work properly. When you get a bacterial infection or a viral infection, your body needs to get rid of those pathogens. So it's been hard to develop drugs to study inappropriate behavior of the complement system. And yet it's the most nasty type. It perforates cells and kills them. So, but multiple new clinical trials with macular degeneration are proving to be very promising. And curcumin has also been shown to reduce complement activation. And so why should you care? It's because with the first, I'm going to show you a picture of the first step in the complement pathway. It's called C1q. It increases 300-fold with aging. So that's why we should care. And 10 to 80-fold in Alzheimer's models. And I'm going to be showing you the pathway, but our lab is very interested in the 5th component of complement called C5a. So here's a picture of that molecule, C1q, that triggers the whole cascade. And all the amyloid proteins can activate the complement cascade. So that's bad, right? So this is a picture of the beta amyloid protein. They wrap around the stem of this complement protein. So it starts here, and you have all these things happen until you get to the end. The end is called the attack complex, and that's what kills the cell. In 17 different trials with macular degeneration, they're targeting this middle part. So they're concerned about like affecting your ability to fight diseases. So they've been injecting these drugs directly into the eye, into the vitreous. C3 is very important, and we don't think it's the best target. But you don't know until you look at the results of the trials that are ongoing. We don't like blocking at the C3, but like I said, there's a lot of trials going on. We're interested in the C5 target because C5 cleaves into A and B, and one of those becomes part of the pathway. A is just floating around, not involved, and we think targeting C5a would be a safe thing to do because it's not part of the attack complex. It's just needed to create the C5b. So what can we do about macular degeneration? So one of the tactics is to inhibit formation of fragile new blood vessels by making antibodies against things that stimulate blood vessel formation. They're trying photodynamic laser therapy that can destroy bad vessels. And leaking blood vessels will allow complement factors to go into the brain. So that's also bad because it can increase your risk for Alzheimer's. See, I'm trying to figure out how am I doing time. Oh, I'm not doing too bad.
You have about 10 minutes left.
Okay, then I may only have 5 more minutes. That would be good. Then you can ask a lot of questions. So, it's our belief that it's wet macular degeneration that overlaps the most with mixed dementia, and that may go together. Mixed dementia has hypertension. Okay, we went over this already. I'm sorry about that. So it's in summary, we have these risk factors for AMD: aging, tau amyloid, oxidative damage, complement activation, leaky blood vessels. And then we have pretty much the same things going on with Alzheimer's disease, especially with mixed dementia, where you have vascular disease associated with hypertension. So there's like 3 take-home messages. Macular degeneration and Alzheimer's are both neurodegenerative diseases with strong involvement of oxidative damage, inflammation, leading to complement activation and accelerated decline with blood vessel leakage. The complement pathway, specifically C5a component, is likely a major drive in both macular degeneration and Alzheimer's. And for macular degeneration, there's 17 trials are underway using injections against C5a. I forgot to put C3 as well. But for oral, we're going to need oral formulations if we're going to be targeting that with Alzheimer's. And these are antibodies, which are not, super expensive. It could cost you $100,000 a year to get treatment. So obviously you want cheap, small molecule inhibitors that target complement and orally bioavailable. Further, complement components in blood may prove to be helpful biomarkers to track disease or risk. For example, going to your doctors and say, could you not just measure my HbA1c or cholesterol or HDL, but could you also measure my C5a? So I'd like to thank mostly Maya Coronio and Steve Verdiner at Cedars-Sinai. They're the people developing this Curcumin technology for eye imaging. Okay.
Great. Thank you, Dr. Frautschy. Gosh, there's so much there to digest, speaking of curcumin. And we have 3 questions, so I'm going to start to look at these questions and ask you questions from our audience. So first one is from Paula, and she asks, how much curcumin is recommended to take daily? And let's just add for prevention or for slowing either AMD or Alzheimer's.
So that's a really tough question because so there's you will see there must be 1,000 different formulations of curcumin. And the best answer I can give you, first let me explain that you absorb it best if you eat it if you take it on fasting, like a half an hour before you eat dinner. So it also is absorbed. So how it's absorbed is, knowing how well you're absorbing it is going to answer that question. So it depends on how you're taking it. If you take it fasting, that helps. If you take it with olive oil, mix it in olive oil, that helps. So how can I best answer this question?
Can I ask you what dosing was used in, let's say, I know we did a study on Curcumin, and we used a certain amount.
Right.
Here, and it showed some benefit in
the dose I did in my trial is 800 mg a day.
Okay.
We're trying, but we're not sure what the optimal dose is to get rid of, because there's different things it's doing. It's what's called a pleiotropic drug. So it gets rid of amyloid, but it also does other things. And it just so happens that too high of a dose doesn't get rid of the plaques. So you don't want to take too much. What we gave the mice is what they eat in India. But then you don't want to take too little if you're not absorbing it. So if you're taking it, I recommend taking it if you can stomach it before a meal, and with a fat, heat it up in olive oil. That's helpful.
Are there side effects, or you said if you can stomach it? No. What are some of the issues?
Well, some people complain about stomach problems, but it's on the FDA's list of it's called GRAS, generally recognized as safe. So it's like as safe as sugar, It's extremely safe because it's on the GRAS list, but you'll go, if you Google it, there are a bunch of curcumin haters and they'll say, oh, it's toxic. And then they'll cite a paper that shows that it's not toxic. So if the FDA, you know how strict the FDA is. If they say it's safe, it's safe. But so they gave dogs like 20 grams. And I personally take like 2 grams a day.
Okay.
But I think the problem, if you're absorbing it well, 1 gram should be enough. So 1,000 milligrams, I think 1,000 milligrams should be enough. But if you feel, the thing is, it takes 10 days to get the blood levels up.
Right.
So the first, you take it for 4 days, your blood is going to have nothing because it breaks it glucuronidates, and it comes out of your urine. And also when your body tags it to get removed into the urine, that version of curcumin does not get in the brain. So you can't just take it one time. You've got to take it for 10 days before it can do any good.
Okay. That makes total sense. Like most supplements, I think they tell you.
All the fat supplements are like that. Vitamin E, E takes 3 months to get in the brain.
Oh, wow. Okay.
Yeah.
All right. And have they done any studies on, how it compares to some of the ocular vitamins that you talked about in the ARED study? Or do we have any information about that?
No, they should do that. I mean, if I were doing the trial, I would use what you can get over the counter, and then with and without curcumin, that would be a good trial to do. The one that's ongoing, maybe they are doing that. I have to look it up.
Yeah, that would be interesting.
But that would make sense because usually when you do a trial, the people on placebo have to use what's the recommended treatment is.
Right.
Yeah. So probably, so that we'll look into that.
All right. Terrific. Let's get another question here from Hope. She says, is there a lower incidence of AD or MD, in other words, macular degeneration and Alzheimer's disease, among people who eat a typical Indian diet?
Yeah. I'm not sure about macular degeneration. I have to look that one up. But I can look it up and then you can email people. But Alzheimer's disease, India has the lowest incidence of Alzheimer's and Parkinson's in the world. And so the curcumin haters said, another interesting thing is that as big cities in India are getting like McDonald's and junk food and stuff, the risk is increasing in those cities. But in the rural areas, the risk is still down. And then the curcumin haters say, oh, but that's because ApoE4 is lower in Indians. So that's the major genetic risk factor that most people who have Alzheimer's have that. But the lower risk of E4 is not in the areas that's insufficient to explain this huge reduction in risk, because it's not necessarily in populations that have low E4.
Okay. What about, Nancy is asking, is blepharitis associated with AMD?
I don't know. Sorry.
I don't know either.
Yeah.
Sorry about that, Nancy.
It usually inflammatory diseases are just going to put you at risk for any of the above. Yeah.
Okay. So
if
somebody had it. Oh, go ahead. I'm sorry.
Go ahead. I just think that inflammation needs to be treated, but how to treat it, it's kind of rhetoric, because we know bad inflammation causes disease, but what is the best way to treat it? Ibuprofen, taken 2 years before, in the years that you're at risk, like early 60s, taking it for 2 years, it seems to do a pretty good job of preventing Alzheimer's. But taking ibuprofen when you're at moderate or late disease can make it worse. So there's no like recipe for like
Right.
helping your inflammation, not yet. But curcumin's pretty good. If you absorb it, it's pretty good at correcting inflammation. So not ibuprofen gets rid of it. It lowers the microglia, but ibuprofen doesn't clear the amyloid. So it doesn't have that pro-inflammatory effect on healthy inflammation. So we need to find more drugs like that. Since pretty much every disease involves that, like it's called resolution, to complete the inflammation so that it clears, does its job, and then it goes back to baseline. That's called resolution. So that's the way in every age-related field, is to get the inflammatory cell to resolve the problem.
Henry's asking about when you're asking for the C5a test, is that the only additional lab to request?
Well, we don't have that test yet. That's what we're working on.
Okay.
So
all right.
We think so. So we're interested. So C5 is super low, like 100-fold lower in the brain than in the blood. So if you just measure for C5, you're going to have an enormous amount, and it's not going to tell you a lot. What you want to know is what got into your brain or your eyes, because getting it into the brain means your blood-brain barrier is breaking down. So if you have C5 in the brain, so how do you just look at the blood and know what happened in the brain? How do you know that you got C5 in the brain? Because it shouldn't really be there. And so the way you do that is you take the blood and you look for these vesicles that are dumping out from the brain, and they should have stuff that brain has in them. So we're looking for, we're developing a methodology to identify C5 in brain vesicles that come from into the blood. Does that make sense? Does that make sense?
Yeah.
Yeah.
Yeah. Yeah, it's kind of like, would you put it along the lines of how we measure A beta in the blood? Is that a different process?
Yeah.
A beta and
yeah, we're developing an assay that is going to be able to be extended to any clinic, any lab. So we're trying to simplify it because as these technologies are being developed, they're quite time consuming. They're not practical for labs to do it, because they might take, you might have to extract this, do this, many steps, like 100 steps to get to that, to measure that. But we have been developing a new assay that's quite quick and easy to identify the brain-derived vesicles in the blood.
So if somebody goes to their ophthalmologist, their retina specialist, for example, and they get an evaluation, I confession, I go to one, and are they able, on a normal scan, they do all the OCT scanning, they take pictures of the eyes, they do fluorescence. Are they able, when they do the fluorescence, to even see anything that would indicate that amyloid is present in the retina? This is basically a question from some of our a number of our
so the company that's working on this is Neurovision, and I listed him as Steve Bergener, and he's working with Cedars-Sinai to sort of bring that technology to the optometry office. So one thing I wanted to mention is that they can already see drusen, and that's like indicative, but the important thing that a lot of people aren't like noticing is that the beta amyloid and tau often occur outside the drusen and is indicative of Alzheimer's. And
in
so
the eye, because I know they've done correlations, right, between amyloid in the eye and Alzheimer's disease, right?
Right. So the people in Cedars-Sinai, they have been studying this really. They have like 15 papers on it. Where the beta amyloid and tau are in association with drusen, because they're seeing it in layers outside the drusen. It's in the drusen. All the amyloid proteins are in the drusen, but the people with Alzheimer's have the beta amyloid and the tau outside the drusen as well.
I
So
see.
it's too early, the technology, but there's a lot of people very interested in it. It's interesting how it's evolved. Like 10 years ago, everybody, the naysayers, oh no, I never saw amyloid, and it's non-specific. And now everybody's on the bandwagon. Yeah.
Well, there are other diseases, right, that have amyloid pathology, right? It's in diabetes, it's in amyloidosis, right?
Right.
Period. In your kidneys, wherever. Are these related types of amyloid to what you're finding in the eye or in Alzheimer's, or is it different?
Yeah, the amyloid and tau are identical, but the macular degeneration, you have at least 5 amyloid proteins, including amyloid and tau. You have vitronectin, transthyretin.
Right.
All of those things are in the eye.
And are those other types of amyloid, are they considered a risk as well, or is it just the specific amyloid in Alzheimer's disease?
I think that they're all a risk. They behave similarly. That
Okay.
yeah.
Yeah.
And
Sorry,
also
it's like more questions.
Okay, so
Yeah.
anti-amyloid drugs, they're gonna get rid of all the amyloids because they're targeting the beta pleated sheet. They all have beta pleated sheets.
Got it.
And they all equally bad. The only thing different about them is that different diseases have different amyloids, except macular degeneration has all the amyloids.
Such good news for those with that problem.
Yeah.
But and someone asked a really good question. Can you just take turmeric, or should you be taking curcumin either as pills, or one of our viewers said they actually have a curcumin eye drop in the UK. So, you know, what's the formulation, or what form of curcumin should people be looking at to take?
So, whatever you do, you need to be consistent with it. So for me, it's like practicality. Some people will juice turmeric. They'll buy turmeric at Whole Foods or wherever and juice it. But if you wanted to go that route, you'd be better off sauteing it, because the oil will extract it.
Okay.
But then you have to do it every day. So yeah.
So you really do need oil, like an olive oil, to help it get absorbed. Is that the point of that?
Yeah. I eat curry every day, and I take curcumin every day. But because I don't know the answer to that question, so I'm killing 2 birds with 1 stone. Okay. But no, I eat curry every day because I love curry, not because of any other reason.
Okay, so there's another question here. This is I'm going to pick one of your questions, David Michaels, but could observations of the macula indicate how well a therapy is working to slow disease progression? And I'm thinking you mean Alzheimer's disease, but let me know if
yes,
you need
they're interested in that. There are people doing trials to see if that's true.
It would revolutionize so much of what we do. It's much easier. Well, I don't know if it's easier, but like, you know, you go to the ophthalmologist, you look in someone's eye, you don't have to put your whole body in a scanner. There's just, you don't, you know, it just seems very, at least another way to increase accessibility of tests, which, you know, to get an amyloid scan costs thousands of dollars.
Yeah.
And it's, you know, they're not done regularly on a clinical basis. So it sounds like this would be a great way to potentially screen for Alzheimer's risk.
Yeah, if people are doing turmeric drops or curcumin drops in the UK, you'd want to image their eyes, right? See if it's just clearing all the drusen out. That
Yeah.
would and you can look at the so their amyloid will already be lit up because you added the drops. Curcumin's fluorescent. So all you do, if they've been using drops every day, you should just recruit them and see see if it's reducing the deposits.
That would be fascinating. So are you keeping a checklist of all the studies you need to be doing?
Yeah.
Okay.
Well, yeah, and I'm not an eye person, so the eye people that we work with are neurovision and
Yeah.
the Cedars-Sinai people.
So they're not doing this at the Stein Institute?
No.
Okay.
But they discovered a lot of the stuff about the human eye macular degeneration from eyes from USC. Carol Miller, she gave them eyes. Sounds gross, right? But then they replicated it in Alzheimer's models, which must have been so hard. That's Maya Coronio. She's like brilliant.
Are there other diseases that are correlated with macular degeneration or has that even been studied? So we know Alzheimer's. What about some of the other amyloid or beta pleated drugs?
Not that I
diseases?
know. Not that I know. I mean, we suspect that cerebral vascular mixed dementia is best correlated. And then Alzheimer's disease is well correlated, but maybe because of the distribution of the amyloid, it has something to do with why it correlates better with macular degeneration than the other neurodegenerative diseases.
Okay. So I think it would be helpful to talk about, somebody asked about, does amyloid show up in MRI brain scan? Do you want to answer that question?
So no. So MRI, it shows the structure and shrinkage and the ventricles. When the brain shrinks, the ventricles get big. So you just see anatomical structural things. You can't see plaques. To see plaques, you have to do what Linda was saying, these expensive scans that use radioactive probe that binds either beta amyloid or tau. So you can, if you have a bunch of money, you can go get imaged for amyloid with this radioactive probe or tau, and then spend twice as much to get a tangle scan. Yeah. And they're not perfect, but they're really helpful for clinical trials. They're essential for clinical trials. So you can track in live patients how well the drug is working. So that's how we know that the vaccine cleared plaques. The amyloid vaccine clears plaques in people very well. And without those scans, we wouldn't be able to track that until they die.
Right.
Unfortunately, it doesn't have a great effect on memory. It's like very minimal. It gets rid of the plaques, but it doesn't improve memory much.
Right. That's one of the things that has been coming up in the research. It doesn't really correlate with memory.
Right.
Yeah.
And we've known that since I got into the field in the early 90s. We've known that. Bob Terry at UCSD, every single Alzheimer meeting, Society for Neuroscience, he'd get up there and said, because he's a neuropathologist and he counts synapses with an electron microscopy. He's like, amyloid doesn't correlate with memory loss. Tau doesn't correlate with memory loss. Only synapse loss correlates with memory loss. And so we've known that. So that's why I got into the field, because I found it so intriguing. The way scientists are discussing things and they disagree, and the field seemed to go really fast early on, but lately I don't feel like the field's advancing fast enough because people have their pet hypothesis and they're not discussing things as well as they could.
Well, this sort of raises the idea of, which has been coming up in the field of, should we be focusing on amyloid or how much should we be focusing on
Well,
amyloid.
we absolutely shouldn't be only focused on amyloid. So we know amyloid causes disease. The Alzheimer's disease is defined by amyloid. If you don't have amyloid, you don't have Alzheimer's. So that's you have to focus on amyloid, but you get amyloids in your 30s and 40s. And then it causes all these irreversible things, like the oxidative damage is irreversible. So if you got rid of amyloid, you would already have oxidative damage from tau. And just blocking amyloid is not going to help you at all with the tangles, because the amyloid gave you tangles, and the tangles are causing oxidative damage. So it makes sense that getting rid of amyloid isn't enough. But then it looks the trials with tau are like not going well. None of them are working. Yeah, so
That's
it's also not. It's getting. You have to think about the whole cascade, and you've got to find pleiotropic compounds that target more than one step, because it takes decades to. We don't know what causes you to convert to starting to have memory problems, like it's called mild cognitive impairment. And then what causes conversion from mild cognitive impairment to Alzheimer's? We don't know any of that. And without knowing that, it makes it hard to find the right treatment.
If you ever look at a brain that's been, you know, a brain with full-blown Alzheimer's, it's so shrunken and it's so undersized. There's no way cognitively, and as a neuropsychologist, thinking you're going to come back from that.
Right.
You've lost too much brain. You've lost too much % of your brain and the connections, and you're just not going to come back from that, even if you get rid of the amyloid, you know, so. But
you might be able to go a long way, because you can function quite well on a few neurons. Like
that's good to know.
There's a case in England where this math PhD student got his, he was a math guy, and he volunteered to do some imaging trial, and he found out like he had half a brain. So this was published in the 50s in Nature or Science. He had half a brain, but he's like was brilliant. So I mean, it is bad, but we should be able to prevent this stuff.
Yeah, that's, I think, where we need to go, so that that damage doesn't accumulate, right?
And I think MCI is key. So we should be doing all the trials on MCI, but the trials on MCI are $100 million a drug per dose. So that's because the reason why it costs so much is that it's hard to power it, because you can't predict when they convert. And there's all this statistics and stuff. And so that's really messy. We don't know how to predict conversion.
Right. It's true. We've done studies of MCI at UCLA, and you have to follow them for years to get enough people to convert. And that means people have to be invested in these studies. They're very expensive to run.
And a lot of people convert the back way. They go back to they go back to normal. It's a really noisy. And we need to design trials better. But
gosh, Sally, it's 4:01, and I know people keep asking questions. Can I ask you just one more on behalf of I think it's Arlene is asking about, and she asked if you can mix like take your curcumin in hot tea, but I'm going to ask you, is there anything you shouldn't be taking it with that could affect its viability or effectiveness?
You should not put it in water. It degrades. You won't digest it in water. So
don't take, what about when you drink it? Don't drink it with water?
Well, you could do it in whole milk. The Indians often put it in whole milk.
Okay.
Yeah,
that's
warm whole milk, but I would add olive oil to it.
Okay. All right.
But it's very hard to steam it. If you like steamed milk, the curcumin will prevent the bubbles from forming. I've tried it.
Okay.
But if you've ever been to Espresso Profeta, have you guys, they have the most amazing turmeric latte. It's like to die for. So go to Espresso Profeta and order turmeric latte.
That's right here
They do
on
it
the
right.
west side of LA.
Yeah.
Yeah. I can't thank you enough for being here today. Obviously, we could keep going, but this is fantastic. Want to answer Diane's question. Yes, this video will be available in a few days on our website at the Longevity Center. Just Google us, UCLA Longevity Center. You'll come to our website and you'll see a lot of our prior talks. Certainly, while you're there, if you feel like making a donation to us so we can keep having these great conversations, please do so. But in any event, we so appreciate you being here today. Dr. Frautschy, this was just a terrific presentation and so informative and illuminating. So I can't thank you enough for being here, and thank you to our audience today. So have a great weekend and keep enjoying those lattes, those turmeric
Thank you so
lattes.
much. Yeah.
All right, take care.
Thank you so much.
All right, bye-bye.
Bye.
Bye.